And thank you for inviting me for this webinar series. I'm really pleased for this opportunity to show what we've done recently for a white paper and the instruments that are most applicable for this type of research. So yeah, that should be my screen coming up there. And I will minimize this one. I work at XENOCS, and we are a small company with the headquarters based in France. And we provide this instrumentation for small and wide angle X-ray scattering that Michael just said that they have in the basement at KU, both in the basement actually. So in the first part, I will introduce what small angle X-ray scattering can do in the fight against the current and future pandemics. And in the second part, I will talk a little bit about which instruments are most applicable for this type of research. And there is opportunity to ask questions after the scientific part before we continue with the commercial break. So I don't think I need to spend much time on this pandemic that we're in. So here in Europe, we're in the second.
Sorry to interrupt, Isja, yeah. But are you intending to already share your screen with us?
Ah, is it not shared?
No, not yet.
There it is, I think.
Perfect, thank you.
Thank you, Kasia. There's a huge impact on, of course, the people that gotten ill and are hospitalized, but also everyone working from home, maybe also homeschooling kids. And the impact there is on the economy, and that makes it crucial to tackle this pandemic as fast as possible. And today I'll share a little bit about how SAXS can help for this. So what are we up against here? This current pandemic is caused by the SARS-CoV-2 virus, a 100 nanometer virus, with this characteristic spike proteins that it uses to enter host cells. In the host cells, RNA is replicated, and the host produces proteins and new virus particles that are then expelled again and can infect new hosts. There's basically two approaches to combat the virus, and that is the development of a vaccine or the development of treatments. And both follow generally this process, which starts with target identification. So it's basically the characterization of parts of the host cells or the virus that can be drugged or targeted with a vaccine. Then lead finding starts, and in this phase, they start finding which moieties or molecules can interact with the targets. After this is decided, we go on to drug delivery. So the platform is decided that can bring these active molecules to the site in the body where they're needed. And then the formulation stage, the exact combination of all the components of the drug are thrown together. After this, the clinical phases start, in which the medicine is tested in humans, and there's manufacturing. And in the top of this picture, you see this timeline, and you can see that this is a very lengthy process. 10 years is nothing. And you can also see that this is an extremely expensive process. In the background of this picture, you see that, that we start with a very high amount of compounds. But towards the end of the process, there are only 1 or 2 compounds that enter the market. And And because of these high costs, especially in the end of the process, as well as the high attrition rates, it is really important to characterize the drug delivery platform and the formulation fast and thoroughly in the early stages. And SAXS is actually an excellent tool to do that. So what are small and wide angle X-ray scattering? Well, in a SAXS experiment, we shoot X-rays onto a sample and we collected the scattering on a 2D detector. The scattering intensity is then plotted as a function of the scattering angle. And this scattering angle is inversely proportional to the probe length. And this means that when we have a large sample to detector distance, we collect small angles, whereas where we have, so we look at large structures. And this is basically what we do in SAXS. So we can look at particle size and shape, for example, as well as interactions. With WAXS, wide angle X-ray scattering, the detector is close to the sample and we collect the scattering from wide angles. So we are looking at smaller structures. So this can be, for example, the liquid crystalline structure or pi-pi stacking. Both these techniques can be done with the same instruments and even be done simultaneously if this is desired. What can SAXS and WAXS bring? Well, this is structural characterization in the range of 1 to 300 nanometers. It can give information about size, shape, internal structure, particle interactions, as well as order and orientation of samples. Various sample types are possible. So this can be gels, but also liquid samples or powders. The results are statistically relevant with just one measurement, which is in contrast to, for example, microscopic techniques. You can do measurements that are sensitive for just the surface, or you can measure in bulk. And it's easy with this technique to measure in situ or in operando. Furthermore, there's basically no sample preparation required. And this is important because it means that you can measure in the natural or native state. So with SAXS, you can have a look at the virus as a whole. You can, for example, look at capsid size and shape and the self-assembly and melting behavior under different conditions, such as, for example, variations of pH and ionic strength. And you can do this with or without the cargo. And this can be either the natural cargo, so the genetic material, or artificial cargo. You can also have a look at the genetic material, and this is actually efficiently studied with SAXS due to its order, for which SAXS is very sensitive, and because of its phospholipid backbone, which gives high contrast. You can, for example, study solid fluid phase transitions. You can quantify the amount of ordered phase. You can look at ejection properties and bending and packing behavior. You can also have a look at structural proteins and non-structural proteins as well, as we'll see later in the presentation. But it's difficult to find druggable targets in the virus as a whole. So let's zoom in into the individual proteins. So this coronavirus has envelope proteins, of which the function is actually not fully elucidated. The spike proteins, which I've mentioned before, membrane proteins, and nucleocapsid proteins. Then there's the genetic material and actually a lot of non-structural proteins as well. Now, this is a picture you've probably all seen. This is a cryo-EM picture that was published only 2 weeks after the full genome was published, which is extraordinarily fast. Now, there are actually 2 big contributions of SAXS for electron microscopy. First of all, because the sample preparation of cryo-EM involves fixating the structure by basically freezing it onto a grid, the this means that you fix the structure in one conformation, and this does not necessarily mean that this is the natural or the active conformation. But SAXS is a solution structure, so you can confirm this with SAXS before you go on to the elaborate refinement of the 3D structure. Secondly, this S protein is densely glycosylated, and these flexible loops, which are very important for its function, they are easily modeled with a low resolution technique such as SAXS. So now we've seen one of the targets, the spike protein. Let's have a look at possible leads. So this is an example that is recently published in preprint. This is a possible treatment for COVID. It's administration of antibodies, and the ones here are synthetic antibodies. And with SAXS, they could make a model of the binding domain of this S protein together with synthetic antibody 23, and thereby elucidate how the binding of this antibody blocks the virus from coming into the body. So to summarize, in the start of this drug development process, SAXS can give fast access to low resolution 3D models, and it can model flexible and disordered moieties. In addition, SAXS creates input for in silico screening, so for the lead finding part, both in terms of providing a model as well as by measuring binding affinities. And vice versa, in silico modeling can also provide structural details for 3D modeling with SAXS. And finally, as we'll see in the next slide, SAXS can also help with lead validation. One interesting example is this of Ribavirin. And this is a nucleoside analog which is built into the RNA synthesis and creates an inactive RNA strand, which means that the virus replication is stopped. And SAXS has helped to clear up why this trick, which is successful for some other viruses, is not effective in coronaviruses. And basically they found that this is because of this non-structural protein number 14, which basically cuts out, is a repair protein. It cuts out faulty RNA pieces. And that means that it also cuts out this nucleoside analog when it's built in. And using SAXS, they found that they could elucidate these very flexible loops in this 3,5 exonuclease domain, which is responsible for cutting out the wrong DNA. Now, if we take it one step further in the process, we enter the drug delivery stage. And 6 of the 44 vaccine candidates that are currently in clinical trials are RNA-based, and often these are delivered into the body with these lipid nanoparticles. These lipid nanoparticles are filled with the RNA, and they help bring this into the host cells, which then produce some parts of the virus that will activate the immune response, but are not harmful for the body. And in this study from the Formulations group at AstraZeneca in Sweden, they used SAXS and WAXS in combination with some other techniques to study these nanoparticles. So here you see both the SAXS and the WAXS patterns. Using SAXS, they could show the size of these nanoparticles, and they could see that they were coarse shell particles. Whereas with WAXS, they saw that that this is an intercontinuous hexagonal phase, and they could see that there was crystalline cholesterol present, as is indicated with these pink lines. Now, both the shape as well as the size and the internal structure have been shown to influence the expression of the desired proteins by the body. So it's extremely important to understand what is going on inside these particles to optimize them. Next step is the formulation phase, and this is basically a balancing act between making sure that, that for example, these nanoparticles are transported inside the body to a site of action, but also making sure that, that drugs or vaccines don't become too viscous for injection, for example, but also for the manufacturing process. And an important factor in formulations of biologicals are protein-protein interactions, or PPIs. These can be attractive interactions, such as, for example, van der Waals, or hydrophobic interactions. And attractive interactions result in unstable formulations, often of high viscosities, and these are characterized by a negative second virial coefficient A2. On the other hand, this is actually desired for crystallization. So if you want to crystallize proteins for example, for protein crystallography, you will be going after this negative second virial coefficient. On the other hand, repulsive interactions, such as, for example, steric or electrostatic interactions, result in stable formulations, often with lower viscosities and characterized by a positive second virial coefficient. Now, PPIs can be modulated with excipients, and these are additions to the formulations that and can be, for example, amino acids or some salts or surfactants or even polymers. So an example where they studied the second virial coefficient with SAXS is this monoclonal antibody 2. And they used this second virial coefficient as a measure for the PPIs. The benefit of using this parameter is that it can be done at low concentrations, which means that you save material for other experiments. But the results are also valid for high concentrations. So you basically don't need as much material. The benefits of using SAXS and WAXS to determine this virial coefficient is that you will obtain a 3D model at the same time. You will obtain insights into the secondary structure. You can gain information about protein folding and unfolding, and it can be done on a large concentration range, in this case from 0.5 to 170 milligrams per milliliter. And the results are buffer independent. And this is important because this means that you can compare the biologicals' biophysical properties across different formulations. So many of the characterizations that have been done in the previous phases are actually also done in the manufacturing phase. SAXS and WAXS are fingerprint techniques. So this means that they have a very high sensitivity for small differences across samples. And one short measurement allows you to grab just one parameter that you want to monitor and check up on for your process. But the benefit of SAXS is that it gives you a lot more information than just this one parameter. So once you find deviating values, you can analyze the data that you have already acquired. And you can find, for example, variations in volume fractions or different conformations, self-association or denaturation processes going on. And this information will help you to decide if you need to discard this batch or if it's something you can continue with, potentially saving a lot of money. Our biosolver instrument is developed in collaboration with academia and industry to bring all of these features that we've just discussed to your laboratory. So in this type of settings, it's important that measurements can be done in-house so that you don't need to schedule, for example, and that they can be done in high throughput, because as you've seen before, if you're going to screen excipients or, for example, storage and manufacturing conditions, you're quickly going to end up with high amounts of samples. It's also important to that you can use low sample volumes to save sample, and of course, automated data analysis is important. And the biosolver combines all of these properties. So just to summarize, the key benefits of SAXS and WAXS are that it's suitable for all types of samples, whether these are liquids or solids or gels or pastes. There is no tedious sample preparation required, and the measurements can be done in native or near native state. As we've seen, you can use a wide concentration range from below 1 milligram per milliliter to over 200 milligrams per milliliter. And you can do static or dynamic sample manipulations to mimic, for example, your processing, your manufacturing process. Sample volumes of down to 5 microliters are possible, and the measurements can be done in-house. The results are statistically relevant. Measurements can be done automated and in high throughput, and this makes this a technique that is interesting for R&D, but also for quality control and manufacturing troubleshooting. It's complementary to high resolution structural determination tools like NMR and cryo-EM, for example, and it provides a good framework for combination with in silico modeling. So with this, I've come to the end of the scientific part.